Good morning. You're at preview day for biotechnology, and I'm Miss Hermans. Thank you for coming. So to give you a brief overview of biotechnology, it's the idea of manipulating living things to produce useful products for ourselves. So any technique that you would learn to do that, or any idea of how that might work, is something that we teach in biotechnology. So something that we first offered this year is something called Ohlone 2+2. So what this means is any student who gets a B or better in biotechnology for both semesters will get Ohlone credit for their Biotechnology 105 class. That also allows them to advance through a certificate course to receive a biotechnology certificate. You would also start the second year at Ohlone if you were to do that sequence of courses. So not only are you introduced to biotechnology, if that's something you're interested in, but you could also receive college credit and also proceed through the certificate course at Ohlone. So a little bit about the course is that I introduce you to basic safety procedures, so things like personal protective equipment, different techniques that are used in biotechnology, including things you may have done in biology like transformations, how to collect data properly, and doing that properly in your notebook with good record keeping, as well as some very broad molecular biology topics like PCR and things like that. We also review basic cell biology, so things like mitosis and genetics are also going to be reviewed in this course. And so you can see a picture that I put here is of what we typically think of as biotech, which is glowing animals. But really what you're seeing is fluorescing animals, not glowing animals, because they've been transformed with a GFP gene. That's green fluorescent protein. So the expectations for this course is that you maintain a quad-ruled bound scientific notebook that I will evaluate every 3 weeks or so. And I'm looking for good quality with everything completed. Also that you exercise very common safety practices. Things like closed-toed shoes are expected anytime that you're in lab, and I usually let you know ahead of time so that you can bring them or wear them properly, as well as nitrile or latex gloves and safety glasses. The homework can be minimal, but also can be a little bit larger than you might expect. After all, it is technically a college level course. Somewhere between 60 minutes to 2 hours a week of homework. And obviously that's going to be more if you're studying for a test or quiz. And I don't collect notebooks if there's going to be a test or a quiz coming up to make sure that you can use your notebook to help you study. So depending on how well you work in class, you might have more homework, or if you work a little bit slower than most or faster than most, you might not have any. Something that really applies to this class is that you have to have a good foundation in chemistry with dimensional analysis and solution making. So if you can do conversions readily between grams and moles, this might be a very good transition from chemistry for you. And then lastly, because it's a lab-based course, we're doing labs sometimes 2 to 3 days per week. You really do need to be present for the lab and to help participate so that hopefully you learn from those techniques that we're going over in class. So some words of wisdom from me is that a lot of the work is self-paced or directed, meaning I give you the work and you work at your own pace. And then I expect you to obviously complete the work and hopefully it gets done. But if you're not very good at managing your time, especially if you're going into senior year, it may not be a good fit for you just because as senior year progresses, your courses start to slip sometimes. So this could be a very difficult course for you to maintain, especially if you need to keep a C for the college of your choice. I do recommend the course if you're interested in it. It just really depends on if you have the work ethic to continue with the amount of work that is assigned. So some of the skills that you learn, and maybe you learned some of these in biology your freshman year or in any Biomed Academy class, is micropipetting. I do go over that in case you've never learned it before. We do a lot of solution making, meaning you have to do the calculations and making the solution yourself. Spectrophotometry, testing for macromolecules, which I know some biology classes do. We really heavily touch on aseptic technique in culturing bacteria, whether this be on agar plates or in broth culture. We also focus on microscopy and gram staining, extracting DNA from various sources, and various assays as well, like ELISA. And we do heavily rely on gel electrophoresis for many applications after some of our techniques. And you can see some of the content that we covered. It's very broad, so there's a lot of techniques, but there's also a lot of information and content that's in there as well. Things like cell structure and function, mitosis and meiosis related to genetics, along with going over DNA structure with replication, transcription, translation, etc. So unfortunately, if you do not like biology, this is probably not going to be the class for you. And I say that just because, you know, if you really don't like labs, there's a lot of labs in this class. And so you might choose some other course that would better suit you, something that you would enjoy. So I really advise you to think about if this is something you would enjoy taking, or maybe you enjoyed having me as a teacher. But if biology was something you really did not like and you liked something like chemistry better, then maybe this isn't the route for you. So I just want to say thank you for coming, and hopefully there are some students present that can answer any questions you might have about the course. Thank you and have a good day.